I hereby open this academic ceremony in which Christian Nogales Calvo will defend his academic thesis, Network Modules as Novel Molecular Disease Definitions for Precision Theranostics. I'd like to welcome everybody present at this ceremony, the supervising team, which is sitting opposite me, and the members of the Corona, which I will introduce later, and we have 2 Corona members that are online. May I invite you to present a summary of your study and the conclusions of your thesis?
Thank you. Highly esteemed Director, highly esteemed members of the Corona, dear family, friends, and colleagues. During the next 15 minutes, I will present to you part of the work containing my thesis entitled Network Modules as Novel Molecular Disease Definitions for Precision Theranostics. First, I would like to start by asking you, what is a disease? Indeed, the answer to this question may appear quite simple and obvious to everyone. However, once we reflect on it, we realize that current disease definitions follow a last century and outdated organization, where diseases are defined by the organ they appear to influence the most, such as stroke in the brain and COPD in the lungs. One or more symptoms, such as hypertension, or to make matters worse, the name of the third doctor that described the disease, such as Alzheimer's, Parkinson's, and Crohn's disease. This has fundamental consequences by the way we approach, diagnose, and treat diseases. In the current therapeutic framework, we will only detect a disease after this has progressed long enough for the patient to start showing symptoms. Only then a diagnosis will be made, and if necessary, a treatment will be applied to the patients. These treatments are usually symptom-based, where a drug is prescribed, usually chronically, to slow the progress of the disease. The goal is thus to improve the life of the patients, but not curing them, because we don't actually know the causal mechanism of the disease. Moreover, these pharmacological interventions usually follow the current therapeutic dogma of one disease, one target, one drug, that obstructs innovation and fails to report patient-relevant benefits. The immediate consequence of this is that current pharmacological interventions are symptomatic and highly ineffective, resulting in high numbers needed to treat. In other words, many patients need to take one drug to prevent one single patient-relevant negative outcome. For instance, the top 10 highest grossing drugs in the US failed to improve the conditions of between 3 and 24 patients. In some extreme cases, such as the cholesterol-lowering drugs statins, even though cholesterol will be lower for all patients, only 1 out of 50 patients will have their patient negative relevant outcome, a stroke or a heart attack, prevented. It is thus quite clear that we need a new way to approach disease, and new mechanism-based disease definitions are of utmost importance. In 2007, the first human comprehensive disease network was published. In this network, we can see diseases represented as nodes or circles, and they are connected with each other when they share a risk genetic association. Moreover, the size of the circles is proportional to the number of disease-associated genes. Because of this, in this network, we can see how diseases interact with each other, and more importantly, have a better view of the causal genetic origin. If we implement the current organ view of current organ-based disease definitions, we can see that these diseases rarely cluster in homogeneous clusters. We can mainly see 3 homogeneous clusters: a cluster of oncological diseases, a cluster of neurological disorders with risk genes mainly expressed in neurons, and a cluster of retinal dystrophies with risk genes mainly expressed in the retina. The rest and the majority of diseases form multicolor and heterogeneous clusters, meaning that our current organ-based disease definitions are misleading and prevent innovation. Importantly, the genes linking these diseases together in these clusters are the potential lead towards new disease definitions and disease modules. But what are those disease modules? Basically, it has been observed that in a human protein-protein interaction network, proteins related to the disease tend to interact closely with each other, creating disease modules. We can incorporate the genetic information of the human disease networks, as well as other disease information, such as comorbidities, to start building these modules. Those genes will be used as seeds, and they will be the starting point to start building our module. We can map these seeds to human protein-protein interaction. Then we can explore their neighborhood, eventually extracting the new disease module and new disease definition. Disease modules will thus be the causal mechanism of complex diseases, where multiple components within a network are affected. These networks are also heavily influenced by an individual's lifestyle and exposome. However, they are not the only type of diseases that we may find. Rare diseases, for instance, are usually caused by a single gene or protein instead of a whole network. So, now that we have defined diseases mechanistically, how can we apply mechanism-based treatments to these patients and to these new disease definitions? Well, in the case of rare monogenic diseases, the answer is more straightforward. Rare monogenic diseases can directly be treated with small molecules directly targeting the affected gene or protein. It is only those cases where the current therapeutic dogma of one disease, one target, one drug appears to be valid. However, and thanks to the ongoing and recent developments in gene therapy, in those cases where the disease is strictly caused by a single affected gene, gene therapy presents itself as a much better curative option rather than chronically treating those patients with drugs. However, in the case of complex diseases and complex networks where multiple components within a network are affected, what will be the best way to approach the disease pharmacologically? Here's where the concept of network pharmacology comes into action. Network pharmacology approaches use two or more drugs targeting the causal disease mechanism in key components of the network in a highly synergistic manner. Importantly, we must not confuse the concept of neuropharmacology with the more widespread combination therapy, where drugs are combined, acting symptomatically on unrelated targets, but they do not target the causal disease mechanism of the disease. Also importantly, because in neuropharmacology approaches, the drugs are targeting key components within a network, we can substantially lower the dose of each individual drug and still reach the same therapeutic effect, or even a greater therapeutic effect, that monotherapy and combination therapy, while at the same time reducing the side effects of each individual drug and possible unwanted drug-drug interactions. Now that we have this strategy, redefinition of diseases according to the causal disease mechanism, followed by a mechanism-based intervention, has been applied to one heterogeneous cluster within the human disease network. In the original version of the human disease network, a cluster of heterogeneous diseases, cerebrovascular cardiometabolic disease phenotypes appear, including diseases such as stroke, hypertension, heart failure, dementia, and Parkinson. The fact that they appear together, linked by shared risk genes, suggested that they might share a common underlying pathomechanism. Indeed, several genes related to reactive oxygen species dysfunction and signaling signaling, currently separated fields, appear to link these diseases into one joint network, ideally suited for the redefinition of diseases. Therefore, To start building the disease model for this cluster of diseases, we started from a strictly validated ROS and CKGMP proteins. Those we mapped to the human protein-protein interaction, exploring their neighborhood, and eventually extracting the ROS CKGMP disease model. We compared two different local methods that yielded similar results. A first neighbors method that controls for highly connected proteins, and the diamond algorithm that assign proteins to the disease model according to their connectivity significance. Both methods yielded a NOX4, NOX5, NOS STC intervention module ideally suited for a neuropharmacology approach. Indeed, with ROS overproduction, CKGMP signaling becomes disrupted, leading to disease. We can redirect the pathway to a healthy status by combining drugs that inhibit NOX, inhibit NOS, and activate STC. Indeed, in a wild-type mouse model of ischemic stroke, the suggested drug combination inhibited NOX, NOS, and activating STC was neuroprotective in a highly synergistic manner, decreasing infarct size, but also stabilizing the blood-brain barrier, improving neuromotor function, and increasing overall survival. Nevertheless, not all patients within this disease cluster will have developed the disease the same mechanism. The challenge is thus to develop diagnostic strategies to select those patients and only those patients that have this causal disease mechanism. Then we can treat those patients with a mechanistic approach, reduce the number needed to treat ideally to 1. In other words, all patients will benefit from the therapeutic intervention. In hypertension, one of the diseases within this disease cluster, we detected a subgroup of patients with high levels of the ROS-forming enzyme NOX5. We hypothesized that those patients have developed this disease through this causal mechanism, and they're potential candidates to a neuropharmacology approach intervention such as the one discussed too. In conclusion, the ultimate goal to redefine diseases according to the causal mechanism is to move from a current symptom-based therapeutic approach to a more mechanism-based approach. Ideally, once the disease starts progressing, we will identify the risk before the patient starts showing symptoms, providing an early diagnosis. We'll select those patients with those risks then and apply a mechanism-based intervention, redirecting the pathway to a healthy status and ideally curing all patients. Thank you for your attention, and I would like to give the word back to the director.
Thank you very much for your clear presentation. The opposition will be opened by Professor Dekker. Professor Dekker was chair of the assessment committee, and he's professor of clinical data science at this university. Professor Dekker.
Thank you, Prorector, and thank you, candidate, for this excellent presentation and very nice thesis. My congratulations on this great work, and also would like to extend that congratulations to your promoting team. Having said that, I'm intrigued by your thesis, but I also have some questions, and I would like to start by asking one of your paranyms to read out proposition number 8.
Network pharmacology will extend even to cancer, enabling non-toxic targeted cell arrest and allowing the immune system to take over again.
Please use a microphone.
Yes.
Yeah. You have to stand. Yeah.
Network pharmacology will extend even to cancer, enabling non-toxic targeted cell arrest and allowing the immune system to take over again.
Thank you very much. Also in your presentation, cancer is not at all in that combined disease area, if you want. So what is your evidence for this proposition?
Hi, esteemed professor. Thank you so much for your kind words and for your question. Indeed, we don't mention cancer almost at all during in my thesis. However, at the end, in the last chapter, we start to see like some potential approaches where they use mechanism-based treatments to cancer too. So I think like right now in this ROS cyclic GMP cluster is quite ideal for this approach that we have used. But the next step forward will be to validate this approach in other diseases. We think also cancer is ideal because we can have a very good view of the driver genes of cancer. And then with these driver gene mutations, we can also explore their neighborhood. And then ideally find potential drug candidates where we can slow the growth of these tumors.
Right, but that requires a lot of knowledge about the cancer. And cancer, if anything, is changing continuously, right? So there's if you think about cancer, there are many different cancer inside one cancer, and they all change as a result of internal and external pressures. So how are you going to learn those networks that you need?
So right now we're actually working on two cancer tumors. One is a diffuse intrinsic pontine glioma, and the other one will be acute myeloid leukemia. So we're starting with low complexity tumors because less modules we think will be acting on those tumors. Sort of just also to validate the principle here. And once we use this approach in low complexity tumors, we think we can expand it to other more complex tumors. So for instance, in diffuse intrinsic pontine glioma, it's a pediatric brain tumor, so not many, we anticipate not many driver genes will be involved. So there will be like less modules active, and therefore it will be easier to establish the concept here.
Right, but how do you, you can start from the genes up, right? I understand that. But really what's happening is a much more dynamic process, right? Where things are, I think your whole thesis about it, it's a network, it's a complex system, things influence each other. So will you determine in such a dynamic environment cause and effect?
Well, the idea is also to these modules also in cell lines from the patients once we have the driver genes, and for that we're compiling all the literature, build the modules, and then from cell lines from the patients, we would like to also validate the pharmacological approach that we can extract from these disease modules. And then we will see if we can inhibit growth or slow the growth or even kill some cells with this approach. And then maybe we can also expand into the clinics too.
Well, what do you think about the following, the following approach, which we are taking is that, if cancer is such a changing system, and as a physicist, if I want to understand a system, the best way to do that is to kick it and see what happens, right? To either, give a drug, or in the case of radiation, create a truckload of reactive oxygen species and see what happens, right? See how the tumor reacts. What do you think about that approach?
So you mean inducing the mutations on cell lines and see what happens if the cancer develops, or?
Well, in cancer you would have, as you say, 5 maybe different treatments, right? You could be radiation, you could have surgery, and of course a whole bunch of pharmaceutical agents. Now, to understand what will work at that time in that cancer in that patient, right, requires another knowledge about that. Right? And so one of the ways to determine that knowledge is to give a bit of therapy, see what happens, give a bit of other therapy, see what happens, and slowly try to identify where the tumor is going. We think that will work and that your networks will help us there.
I hope. The whole point of the networks is to sort of anticipate what will be going in those patients. We will also would like to have a look at the genetic, because once we identify modules in general, we would like to also have a look at the genetic makeup of the patient to see what genes are affected specifically in these patients too. And then also the treatment will be personalized to each individual patient.
Right, but of course cancer is similar to the patient, but it's also changing continuously. So that's an interesting topic. let me ask you another area, right? As a data scientist, I'm also interested in, what causes disease and how should we treat that. I have made a picture of, and which is similar to your pictures, right? Which I show you here. It has these people on there with colors, right? So which of these colors do you think is your genes or your biology? This is a color that, the title of the infographic is "What Makes Canadians Sick?"
Okay.
So what is biology here? Which color do you think it is? What's your hypothesis?
Will it be all of them?
No, it's purple.
Okay.
So 15% of disease is being explained by biology.
Okay.
Right, and 50%, the red things is your life, right? Your income,
Lifestyle.
your social, your income, your education, your social network, and so forth, right? And for instance, we know very well now in the pandemic, right? But if you want to predict if a COVID patient lives or dies, right? The most important question is, are you vaccinated? And being vaccinated is very much determined by these factors, right? Which network, which Facebook pages do you read, what's your educational status? So given that, right, given biology is only 15% of disease, what makes you assume that you can solve this by biology?
But aren't, even though the others are to the diseases like due to socioeconomical issues, wouldn't those also indirectly also affect the biology of the patients? Because let's say,
so you think the choice of a vaccine or not is your biology?
No, that's not. No, that does not. No. But still,
So my question is a little bit about if you, and I totally agree with your thesis and the way you should view medicine. Let me also say that. But I also think that to treat disease, right, as you say, you kind of make the assumption that a drug is the right way to keep people healthy, right? It may be that we need to educate people more, give them more money, right?
Yeah.
What's your, and if you agree with me, because I think you agree with me, right? What is needed to get there, you feel?
I mean, then if we want to educate people to trust science, we need to spread science and be more engaging with people so they can actually trust scientists more. Because, for instance, in the issue of vaccination status, and people not wanting to get the vaccine is mainly because, in my opinion, because they don't trust the technology behind it. They think something that is completely wrong about it. So that we will change, I think, by educating people and being more open about the science and what we do, and maybe explaining better what's going on behind, for instance, in this case of the vaccine, because I think in some cases people are completely misleading about what the vaccine is actually doing in your body, especially with these new mRNA vaccines. I think some people are actually scared that they might in some way affect their genome, and that's why they actually don't like this kind of vaccines either.
Right. So I like that you're taking a more societal approach, that you could also go very much data science approach to that, but we'll discuss that perhaps at another time. Thank you, and I'm going to give the word back to the professor.
Thank you very much. The opposition will be continued by Professor Schmid. Professor Schmid is Professor of Molecular Pharmacology at the University of Groningen. Welcome, Professor Schmid. We are happy you'll be able to participate online. The floor is yours.
Thank you very much, Rector. And I would like to start with my compliments to the candidate and also to the promoting team, to the supervisors, as I was very much impressed by the work which has been delivered. And I also was very much charmed by the presentation, a very clear presentation you provided here. several of the chapters are obviously already published, and some are in the process of being published. Being a molecular pharmacologist, I would say I'm very much for drugs, and also for the drugs which we have based on lots of clinical trials. So also basic biological studies, translational studies, animal studies, human trials. But nevertheless, you touch in your thesis a very important aspect, as you really try with your novel approach, the Theranostics, you try to overcome the death valley, what we call, from the development of a potential drug target to the real patient benefit. But please explain me the ultimate added value of your approach, as at the end you also, as in the other approach where we most likely tackle bias as we interpret data and collect the data, you also rely on huge data banks, and you sort out a potential mechanism, and then you go for this mechanism, if I understood it correctly. So can you highlight or emphasize the distinction between the classical approach of drug therapy?
Highly esteemed Professor, thank you for your kind words. If I understand correctly, you would like to know how we will select the drugs to these modules?
You can start with that. That's also a good approach. Start with that.
Okay, yeah. So basically what we do is starting from known driver gene, known clinically validated genes that are affected in a disease. From those, we will build the disease module. And then using the data available from drug bank and other drug databases, once we have this disease module, we can look at what drugs are targeting proteins within this module. Ideally, we will focus on our readily available drugs, so we can repurpose available drugs, and we don't have to go through the process of developing new drugs for any of the proteins within this module.
Okay, this I do understand. If I may ask you or challenge you here on this regard, at least what I read in the practical chapters, which I read with with quite a lot of pleasure, for example, chapter 5 and chapter 6, in which you try to translate your theoretical findings to experimental approaches. You purely focus on one signaling box, so on ROS-related NOX5 signaling, which is for me quite a bias or a reduction to the diverse information you get out of the networks. And you also used, maybe I'm mistaken, but you used also quite experimental drugs. So we cannot really, in your approach, yet talk from repurposing of drugs. So please explain me why you focus purely on cyclic, on the NOX, cyclic GMP NOX5 pathway linked to ROS, and why you have chosen this, for example, in chapter 6, for me, quite experimental drugs, far away from repurposing of drugs. Can you illuminate that to me?
Regarding NOX5, it's because in chapter 5, we mainly saw that it was NOX5 and not the other NOXes, the one that was directly connected with cyclic EMP. That was also validated later in the patients with hypertension, where we found a subgroup of patients with elevated NOX5 levels. And then also later in mice, we saw that the NOC in mice with NOX5, when they age, they would get hypertension compared to normal mice that did not have NOX5 in their genome. Yes. Regarding the drugs from chapter 6, so
maybe I can. Would you agree that these drugs you has chosen are not really in the frame of repurposing, but they are also, I would call them still quite experimental. Would you agree on that? And if so, can you explain me why you have chosen the drugs you have taken for your triple approach in chapter 6?
Perhaps they're still like not really approved yet, but I think there are very promising clinical trials that show that these drugs could be soon in the clinics and could be soon be used for patients. So we could actually, I think it makes a lot of sense to use them because the results from these clinical trials are actually very promising.
Good, but you also agree that you are not yet in the drug repurposing frame.
Scope
yes
of
or
my
no?
thesis, no.
Yes or no?
I'm
Just
not,
a short answer.
In the scope of my thesis, I'm not.
Very okay. That's very nice. I would like to ask you, as if I understood it correctly, I see all these beautiful networks and I see all these overlapping themes you presented. For example, also in your recently accepted tips review, congratulations to that. But if I understood it correctly, and it's also stated in one of your propositions, and it's clearly seen in one of the figures in your tips review, that the mechanisms you are analyzing, or that there are substantial overlap in mechanisms. The mechanism you're focusing on, the cyclic GMP mechanism, ROS related mechanism, has quite a substantial overlap to RAS maybe signaling and Wnt signaling, but also, for example, cyclic AMP signaling. Can you explain me why you have given that, the overlap? Can you still explain me, or why you're focusing on this cyclic GMP and have not taken cyclic AMP networks into account, or drugs, to use such drugs, as there are several PDE or cyclic AMP related drugs, PDE inhibitors, beta agonists, or beta compounds, which you could have used in your experimental models, which would be a drug repurposing approach?
We mainly focus on ROS cyclic GMP targets because in the human disease network, which is the starting point to look into the mechanism of these diseases, it was mainly ROS and cyclic GMP genes, the ones affected. However, there might be other mechanisms that also led to some of the diseases within this cluster too. So with this mechanism that we described with ROS cyclic GMP, we think that only we can treat a percentage of the patients. And if things are consistent between NOX5 hypertension paper, it will be only 1/4 of the patients, but we're also planning to endotype more patients within this cluster and see how many of them actually have this ROS cyclic GMP mechanism. Ideally, after that, I think it would be nice to move to other, so to also find out what other mechanisms lead to these diseases too, that they are not ROS cyclic GMP related.
Okay, I'm not sure whether my time is already up. I have to look to the...
I'm sorry.
Oh, it doesn't matter. It was nice. I give the work back. Thanks for the answers. I give the work back to the prorector.
Thank you very much. The opposition will be continued by Professor Blokland. Professor Blokland is Professor of Comparative Neuroscience at this university. Professor Blokland.
Thank you very much, Prorector. Dear candidates, first I would like to congratulate you for this wonderful thesis. There's a lot of work that was in there, and of course also the promotion team that helped you to guide through this topic of network medicine. I know that you have been submerged in this group of network medicine people, and I would like to scratch on the surface of the validity of the model that you use. Mm-hmm. So there are different things that come out of this. So that I was already trying to ask you about the repurposing of the drugs, but My colleague also had some questions, so I would like to stick to one part of the question. So I think repurposing is not so really interesting for drug companies because the patent situation, and when you look at table 2.2 in your review, you see that all the companies make new drugs.
Mhm.
So I do not agree that we can just get rid of all the classical drug discovery. So that's quite a bold statement, I would say. But or do you think repurposing will be the future of drug discovery? Maybe that's the first question.
Highly esteemed professor, thank you for your kind words and your question. In an ideal world, I think repurposing should be the future because small molecules are actually highly promiscuous. So the likelihood to find new targets from already approved small molecules is quite high. However, if we consider the interest of the companies and maybe they make more benefit by developing new drugs, then we dig into not so nice area anymore, indeed. But in ideal world, I would like to see the drug repurposing as the future.
That's good. Maybe you should realize there is no ideal world. But anyways, maybe we could go to, so I think already was discussed a bit about the implication of the network. So I think what you show in your picture is like we want to go to the mechanism and then you would bend the curve and to stay in a healthy state. But when I look at your one of the final slides, I only see that 2 out of the maybe 10 puppets you had there, they were treated. And you just mentioned, well, maybe there's another mechanism. But I mean, I think the model predicts that if this is the cause of stroke, then this should work. But it only works in a subset. So the network model approach does not predict everything. And maybe that also related to an earlier question, what are these other factors that cannot fully be explained by the network model?
Well, we start our network model with a set of genes and proteins, and those are the ones that have been studied the most for an implicated disease. But there are also maybe other set of genes and proteins that have not been so much studied, and they might also lead to the same disease. So let's say we have already defined mechanism A, and this would be raw cyclic AMP for us. But now with this mechanism, we can only treat like 1/4 of the patients. Then we need to find out what this other third-fourth of the patients how they have developed this disease too. So what we might think in my work is like from the human disease network, we subtract those genes that we have used to develop this mechanism and see which ones are remaining. And then from those, we can do the same method again and see whether new modules search, and then from these new modules, find new potential therapeutic options and see if these new modules are actually real and validate them with patients. Yeah.
Okay, I can relate to some extent with you. Because when I was reading your paper, one of the chapters on page 163, say to test the causal endothelial NOx5 hypothesis. And when I read the word causal, I get shivers. So how can you be sure that this is a causal mechanism? What evidence do you base it as a causal mechanism, NOx5?
Well, regarding NOx5, I think we also briefly discussed it before. So we can see the module is the module and will only be a model at the end. So then when we move to patients, we can see that 1/4 of them have this high elevated NOx5 levels. And then when we knock in mice, they also develop hypertension when they age, compared to non-knock-in mice. And actually, it's not in my thesis, but I can also tell you that another disease of this human disease cluster, we have also detected a subgroup of patients with high levels of NOx5 that also match 1/4 of the patients, such as in hypertension. So the data is also highly consistent so far from what we have measured.
But you only refer then to one mechanism.
Yes.
That is causal for the whole effect, for hypertension.
That indeed, that's not right. Yeah, this
So the
mechanism would
network
be,
model would suggest that it is more than one.
yes, yes.
So then I still don't understand the causal, why you use the word causal, because then I would expect that you say these, and these
Okay.
are the causal mechanisms.
Well, it would be causal for this 1/4 of patients. It is causal to cause hypertension, but not for the others. Perhaps it's not the best phrasing indeed. But at the end of the day, it would be causal for 1/4 of the patients indeed, right?
Maybe.
Maybe.
Yeah, yeah, but I've a little bit more. Yeah, a little more time. Huh? Yeah, a more scientific question, a more general question. So when you look at page 162, so you present the data of the NOx5 levels in plasma, and then you see that a lot of stars, and we love stars because that's a difference, and we conclude NOx5 is. Oh, you see the picture, or?
Oh yeah.
You should remember that.
Yes, done. Yeah.
Okay, so we at least see a lot of stars, but when you I think you already alluded to that. So
Oh, yeah.
it's only a part of the people with increased levels. But what would be the conclusion? So I think we all scientists say there is NOx5 is relevant for the, but it's only a part. It's only maybe 20–30% if you consider the overlap. So what should we actually conclude on base of the data as scientists? You already said we should be more careful to truth with the truth of science. So how can we rely science?
Mm-hmm.
So when we make a statement, like if you have a chapter and the title of the chapter is NOx5 is induced uncoupling, but it's only a minor part of the whole group that is related. So how should we actually phrase the title in order to make us scientists more truthworthy?
How would we rephrase the title?
Or do you like the title as it is?
Perhaps could introduce the endotype word in the title. So then I will mention that this is just an endotype of hypertensive patients. So it's only a fraction of the hypertensive patients, the one that would be developing hypertension to this NOx5 mechanism.
Yeah. Okay, but that was just a general remark. Thank you very much for your answers, and I give it back over to the prorector.
Thank you very much. The opposition will now be continued by Professor Prikkaerts. Professor Prikkaerts is Professor of Experimental Neuropsychopharmacology at this university.
Thank you very much, Prorector. Dear candidate, first of all, I would like to congratulate you with this excellent thesis, of course. I would also like to congratulate your expert promotion team, true experts in this field. I learned a lot, very conceptual reviews, big experiments, but a quite provocative thesis. I have the feeling we are a bit hard on you, but I would say see it as a compliment for the thesis. So I'm going to continue on that, and I still want to linger a bit with the repurposing. You've noticed already we have some issues with that. So for the audience, I would like to ask one of your paronyms to read it now, number 7, in the microphone.
Repurposing registered drugs can rapidly address unmet medical needs, outcompeting classical drug discovery.
So drug discovery is dead, long live repurposing. Okay, but that's actually what it says. But I have also a problem. You can only repurpose if you have an existing drug. And I think we have not exploited fully all the targets. I work on PDE4, you know that target. Later on, we discovered there are 4 types, PDE4, A, B, C, D. And now we know that D has 9 isoforms. And we're now trying to make the drugs. As soon as we have them, I can hand them to you, and you can start your repurposing. So I think drug discovery cannot be that, because otherwise they will not be repurposing. What do you think?
Highly esteemed professor, thank you for your kind words and for your question. Indeed, I think there is quite clear crisis in drug discovery because we have this the Eurom's law described that the discovery of new drugs is in constant decline. So that we have to find a new way to tackle this issue anyway. And I think that drug repurposing is an ideal option in the cases that can be implemented. In those cases, with the modules method, we can clearly see what drugs already approved can be used in those modules. Obviously, in those cases that we don't have a well-defined drug to target the disease or the protein, drug discovery is still needed. Although we can also maybe make use of prediction tools to predict whether a molecule that has already a validated, it's like clinically available and has a approved target, could be by predictions used in a different already protein that we might have found a disease and doesn't have any pharmacological option available at the moment.
Actually, I agree with you. Drug discovery is not that successful in CNS, but it could also be related to something else. So do you know the person Paracelsus?
Paracelsus.
Yeah, that's the founder of toxicology, actually. And
The dose.
this is a pharmacology thesis, you should know that.
The dose makes the poison.
Yes, the dose of a drug makes the poison. So it's the dose that determines what the drug does. So maybe the statement should be inappropriate dosing is dead. Long live the right dose. What do you think?
Perhaps still there are not many new drugs being discovered as much as they were like in 50 years ago, let's say. So in that sense, drug discovery is being in a stagnation, actually.
Yeah. I just want to make the point. It's not only the drugs; it's also the dosing.
Okay.
And especially if you don't know exactly the target, because then you are off target if the dose is too high.
Yeah.
So that's actually I wanted to have some nuance in this. So, but let's leave it with repurposing. We discussed this a lot. Let's focus on chapter 2 and 6, and I would like to go to page 215. A very itty-gritty question. I've seen this picture in many of your papers of your group, and I think the mistake is still there. I think it's a mistake. You see NOS causes ROS and NOx causes NO, figure 6.2. It should be the other way around. NOS produces NO, and then the nitration of tyrosine, of tryptophan, and NOx produces ROS, which you measure with DHE. So you have to flip this, I think.
Actually, you might be right, I think. Yeah.
Yeah. Okay. So point of attention there. Good. It's nice that somebody says I'm right. At home they don't do that. Anyway, good. We continue with chapter 6 and 2. Nitric oxide synthase inhibitors are drugs you use. In chapter 2, you describe one. In chapter 6, it's SMTC, and you say it's NOS1 and NOS3, so NNOS and eNOS inhibition, predominantly NNOS, neuronal NOS. And in chapter 2, a very interesting one, I think your supervisor worked with it, VAS203. And according to the figure, it's an NNOS, neuronal NOS inhibitor. But when it's in phase 3 for TBI, traumatic brain injury. But when I check the mechanisms, they say it's actually iNOS, a NOS which is not even described in your thesis.
Okay.
So you have TBI, there's inflammation, so the microglia have increased iNOS, and when you inhibit this, that's good. But you come up with a totally new mechanism, NNOS, and also found it does eNOS. So actually it's a dirty drug in my view, based on my information. So how sure are you it's only NNOS?
Actually, the information I had, there was had like a preference for NNOS, although I might also target the other noses. That at least that's what I understood this drug.
It's good, yeah. But in your figure of 2.4, page 35, the arrow only goes to NNOS, and there should also go one to NOS1 and 3. That's eNOS. And that's my next question, because if it does go to eNOS and you inhibit this one, it could actually be bad, because you will have less NO, less cyclic GMP, and blood pressure could go up. So that's not good even. So any thoughts about that?
Perhaps.
I could help you, but you could actually say that we give them an SGC stimulator to overcome this. We should go for combination treatment.
Yeah.
But then the final question, if I may, or have some time, okay. So I linger with this. So in chapter 3, again, a NOS inhibitor, mainly NOS, because that's your favorite target probably, is part of a triple treatment for stroke mice. Or actually, these poor animals had a stroke, had diabetes, and some even hypertension. I wonder how do they survive? But anyway, but you show that the combination treatment really works. And then you conclude that all 3 drugs together work synergistically. But you only show one experiment, and that's the combination of the 3 of them. To be really sure that they add up to each other, you should do separate experiments with the duos, the separate duos, three duos. And if possible, even a high dose, one single drug. And I have the feeling if you would do these experiments, that the NOS inhibitor actually would not make any difference. You would not see it. That's my feeling.
Okay.
So what do you think?
Perhaps you're right, and we should actually have done those experiments beforehand, before we could actually claim
synergistic effects.
Yeah.
Yeah, but yeah, actually, I was hoping that you were going to tell me this is actually the concept of network pharmacology. That
Yes,
you have to combine.
we have to use lower doses in order to reach the synergy for neuropharmacology to approaches to work. So perhaps it's the dose again. What's
the dose and some more experiments maybe.
Yeah.
But thank you. You thought about it, so I'm very satisfied.
Thank you.
Thank you very much. Give the word back to the prorector.
I now give the floor to the last opponent of this session, Dr. Zanin. He's from the Spanish National Research Council in Spain. We're very happy you can join us online. Dr. Zanin, the floor is yours.
Thank you very much, Prorector. And first of all, let me say that I'm sorry and apologize for the fact that I cannot be there, but as you know, the situation is what it is. And luckily we have technology, so at least we can see each other virtually. And second, I would also like to join my colleagues in congratulating you and your team for this work. I found it dense, quite big work, and also a complex one. Complex in the good meaning of the word, like in complex networks. So something that is not trivial, but is indeed interesting and that has a lot of value in it. I would like to make a couple of questions, especially about chapter 4, because my background is actually networks. So this was my natural chapter to try to understand. So I would like to ask you, first of all, in page around 130, you start by criticizing a little bit pathways, saying that pathways, they are biased, lack important interaction, and do not integrate sufficient information about subcellular localizations, which is quite a strong attack, I would say. I mean, you're almost saying that pathways are useless or not really reliable. But in the next, so in my mind, it was clear, okay, then you move to PPI, to protein-protein interaction networks, because probably they are better. But then you have a sentence in which you say that essentially PPI networks are also biased. And you say, like, for instance, that the centrality is correlated with the number of times scientific publications mention protein representing genes and so forth. This was a surprise to me also because there are a lot of works in network science which have shown that there is a very nice correlation, I don't know if causality, but at least a correlation between lethality of proteins and their centrality in the network. So let's say that my question is a little bit twofold. First of all, then how do you explain the fact that we actually see results with the network, even if you say that these networks are biased? And second, if these networks are biased, then the work that follows is based on this network. So how did you take into account the fact that maybe the data in which you are basing your work is not exactly perfect?
Highly esteemed opponent, thank you for your kind words and for your question. Indeed, I think there are I believe there are limitations with the process we use. We cannot claim that the approach we use is perfect and has no drawbacks. However, even if there are like biases in the network, because there are like, obviously, even from the point of choosing the seeds, some proteins are more studied than others, and that's why they are linked to the disease than others, but perhaps are other potential diseases that we are missing. However, still, even if there is some bias and some limitations in the networks that and the models that we extract, you can still see clear messages that can lead you towards applications out of them. For instance, in chapter 5, the clear message is that it's NOX5 and not the other NOXes, the one that is like inducing the disease for hypertension. So I just think that at the same time, you have to be very aware that the method is not perfect and there are limitations with it that we should also progress towards and try to fix somehow.
But along this line, have you considered the possibility of trying to filter the data from the PPI interaction network? Or, for instance, have you also included data about forecasted or predicted interaction? Have you filtered only according to some criteria? What is your view on that?
To us, more the experimentally validated interactions rather than predicted interactions, because it is clear here that the connection exists and that those proteins are interacting with each other. However, if you move to predicted interactions too, the networks that you extract and the models that you might extract might start to blow up and maybe even too complex to digest for application points of view. Obviously, now that we have this module, maybe it will be a good idea to see if with predicted interactions we can also extract potential new candidates that also make sense that they belong in this disease module, and maybe there are potential also applications for targeting them with drugs, or also for biomarkers to stratify the patients to this mechanism.
I understand, kind of going back, that is not just use this network, but also use the results to go back. That's interesting. Let me make more technical questions. A couple of pages after that, you introduce a metric that is this subnetwork participation degree. That's perfect. And then you say a stringent SPD cutoff value of 0.4 was selected. And this surprised me a little bit in the sense that why 0.4 and not 0.45? Or so have you performed any kind of sensitivity analysis on these results? So why has that value been chosen?
So basically what we do is from this subnetwork participation degree, we can get a view of how relevant the interactions of the protein are within the module that we extract. And we get this value from every protein that we have in the preliminary version of the module before the cutoff. What we have been doing is use an elbow method that sort of give us a cutoff to where no new information is being added. So from that point onwards, we choose the cutoff, and then the other interactions we don't consider to be disease relevant for the disease model that we are identifying.
But let me understand that this cutoff, that is this 0.4, is set by you or is derived from the data?
It's derived from the data, and at some point we use an elbow when the data plateaus, and from that plateau onwards, we don't
that's perfect.
Consider it relevant. Yeah.
Okay, that explains. Okay, I couldn't find that explanation in the text, and that's why I was asking. Okay, I have one last question. More than a question, is not. I'm not asking you for the truth. Is more asking you about your personal opinion on this. from my field, or from my point of view, I always thought that pathways were like the golden through. They are there, and you have to follow that. The pathway are true, and then you can try to do other things, but you have to go back to those pathways. And I see from what you show here that it is probably not right. So do you think there is space for redemption for pathways? That is, what is your view on this? We have to improve the way we construct pathways and we understand them, or are pathways completely wrong and we have to take them away from cell biology? What is your view, your personal view on this point?
You
So-
want to give a very short answer? Sure.
Yes, so I think we have to be aware of, again, of the limitations. We still can improve them. Like, for instance, being aware that some proteins are like in different cellular compartments, and in your mind map version of the pathway, they might not be interacting with each other ever. So we can also reflect that on the newer version of the pathway, and maybe they will be a better guide for future researchers.
Okay, perfect. Thank you.
Thank you, Dr. Zanin. Mr. Calvo, the time appointed for defending your thesis has passed, as you've seen. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
Thank you. Microphone.
Mr. Calvo, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Schmid is authorized to confer upon you this academic distinction in accordance with Dutch university law. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Christian Nogales Calvo, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. You may take photos.
The word is to Professor Schmid for the laudatio.
Dear Christian, congratulations from Anna and me as well. When I prepared for my laudation, I became a little embarrassed. I came across the advertisement for your PhD position, PhD student in recombinant antibodies and diagnostics. Oh my God, your PhD is the perfect example for my warning that our research tries to plan where we go, but after a year, every planning becomes a bit fuzzy. Because we go where the most burning question is, and thus things may change a little. A little? I cannot count how often I said, after you were trained in phage display technology and recombinant antibodies, and you did a great job learning and applying, that just in the moment, some other aspect is a little more burning and interesting. But we'll come to antibodies and phage display. It's not dead yet, it just smells funny. We had some antibodies that work, so let's work with them for the moment. And then bioinformatics and systems medicine came along. First, we thought we can collaborate and let others do for us what we need. However, that was not really the case. The application of systems medicine and network pharmacology for medical purposes turned out to be a subdiscipline itself. Big data are nice and essential, but eventually we have to distill them down to small data, precision data. It's of no help to receive a network with 100 nodes when you want to define a disease and design a new therapy. Thankfully, you understood what we needed and had also a talent for talking to bioinformaticians, learned autodidactically, I would say, certainly not from me. So we slowly created something like applied systems medicine and network pharmacology. Not that I understood what exactly you were doing in phage display. Now I also did not understand what you were doing when coding in R and working with Cytoscape. But still, we operated as a perfect team. I could pretend I know what I'm talking about when dropping buzzwords such as first neighbor, interactome, I don't like Steiner trees, SPD, and so forth. You, however, really developed that methodology and approaches for pharmacology to perfection. Wonderful that this was rewarded, just like week, with a high impact review in TIPS. It's also nice that you took this road by being co-supervised by Anna, who grew along to a soon-to-be professor in Essen. And maybe she assured a couple of times along the way everything will be fine. I have the feeling that this is only the beginning. You are so poised for what is the future of pharmacology and drug development, diagnostics, systems medicine, network pharmacology, that you really hold the future of our field in your hands. There's one more thing. Per aspera ad astra, through hardship to the stars, was always something that I had to think of when you were wearing your NASA shirt. You are obviously a space fan, but we are Europeans. We have to do our European thing. Luckily, there is the ESA, the European Space Agency, and the Astronaut Training Center in Cologne, with a mock-up of ESA's Columbus laboratory in International Space Station. And so laboratory and space come together. When Dr. Nogales will get a private tour once Corona permits.
Really?
Thank you for working with us, being such a pleasant and high-performing team player.
That's my Oh, that's cute. Thank you.
Dear Dr. Calvo, on behalf of the Board of Deans, it's a pleasure for me to congratulate you with the honor you have acquired. I think you have deserved it. You had a little bit of a hard time during the defense sometimes, but I hope you feel better now and really relaxed. And if you think it over, you should realize that you've done very well.
Okay. Thank you.
We come to the end of the session now. So I would like the audience to leave the room after I've closed the ceremony. We stay here to make a picture and just to give some congratulations. So I would like to ask you to start leaving after I've closed the session. So hereby I close this academic ceremony.